All right, so thanks for this opportunity of showing you our work in relation with artificial intelligence in drug discovery. So I'm Victor Guallar. I'm a professor at the Barcelona Supercomputing Center in Barcelona, of course. And also I'm CSO of a spin-off of the center that's called Nostrum by Discovery. I'm actually gonna be basically repeating the talk I gave a month ago in Discovery on Target conference in Boston. So it's largely devoted to illustrate how we are combining molecular modeling and artificial intelligence into facilitating drug development and screening. Let me first tell you a little bit about our center. So we are the National Spanish Supercomputing Center. It's called Barcelona Supercomputing Center. We host Mare Nostrum 5, which is one of the largest supercomputers in Europe. It's quite large and really fulfills our needs. And also, this National Supercomputing Center is a research institution. So we have 4 academic departments that we are conducting research: earth science, engineering, computer, and life science. And I would say it's possibly the first research center in Spain when you take into account the number of money that the center brings in for research and divided by the number of people we are. But even in absolute numbers, we are the second Spanish institution in receiving funding from the European Union. And important projects like the European chip or European quantum computing are being developed today at Barcelona Supercomputing Center. And this is actually our building in the upper zone of Barcelona. It's next to universities area. So it's quite nice area. Also told you that we opened a spin-off around 2016, actually. So it's going to be soon 10 years, where we run a lot of different, all preclinical computational stages like hit finding, lead optimization, specializing a lot in artificial intelligence on how we can apply this artificial intelligence into drug discovery. For example, we've been very successful in working with top 10 like, for example, this year we will finish with 4 top 10 big pharmas. Here you see some of our current and some of the past clients. And for example, we opened offices in Boston in 2025. So we are quite expanding. So the lab, our computational lab in the Barcelona Supercomputing Center, that's in the life science department, it started because we developed this Monte Carlo traditional, let's say, molecular modeling techniques, these Monte Carlo techniques where we could see how, for example, this is a ligand, how it explores the surface of this protein, and how we can, predict the binding region of this protein, of this ligand, how the ligand will enter this center and how it will find the pocket. Actually, this is work that we did with AstraZeneca many years ago. And because of this sophisticated molecular modeling, that's actually why we started the company. We started working a lot with pharmaceutical companies. We can also this is very fancy, putting the ligand outside and seeing how it explores the entire surface and it will binding. It's more useful probably just to find how the molecule will be adapting to the pocket of our receptor. So in drug discovery, what we are mostly doing is finding drugs, which are small molecules that can very nicely fit into the receptor pocket and can make a function. For example, inhibiting this protein for working, or the opposite, or activating this protein to work better, or this enzyme. And of course, once we have molecules, we can actually even grow them. And using these molecular modeling techniques, we can derive new molecules, for example, by expanding new fragments and seeing how these new fragments will be fitting in the pocket of this target. So this was a lot of molecular modeling. This is expensive. It takes a lot of computing cores, multiple hours. So, it cannot be used in a really large high throughput scheme. And what we want is to apply this into a very, very large number of molecules. Let's say that the green curve that we see here are all the molecules that we could sample, or let's say hundreds of billions of molecules that we would like to try to assay and possible. So instead of doing this very large space, we can do a much, much more reduced space, like the one that you see below. If we could do, however, the larger space, we will get always systematically very, very good drugs. But it's just very difficult to assess this larger space. So let's say that if we could really sample these molecules in this vertical orange line, half of the molecules, even if we do computational, which is not the reality of trying experiments in the lab, even if we did computational, we will find possibly half of them are going to be very good molecules, but that's not what we do. What we can do today is like this smaller subset, and there's a very, very low hit rate. These molecules are not really fitting well our target, our prediction of molecular modeling are not good, etc., etc. So what we need to do is to shift this distribution. Of course, we cannot do the molecular modeling of the entire set because it will be too expensive. And what we would like to do is shifting, as you saw here in this moving Gaussian, shifting our distribution that we can do towards better and better molecules. And the way that we are doing this is basically by looking how these molecules will be fitting in our pocket. We can score that computationally with a scoring function that we name it, and we can select them. And of course, we want to do the larger, the more, the better. For example, in 2012, like 13 years ago, typically libraries of molecular drugs, of molecular drugs that we could try computationally, like all these different symbols in here, the star, the circle, etc. All these different molecules were accounting for around 1 million molecules. Then 2015, we have libraries of around 3-4 million molecules. The first billion-like library so 1 billion molecules that we have sitting in a hard drive that we can interrogate computationally started in 2020. And now we have libraries of around, actually 2025, we have like around 70 billion molecules that we can interrogate. And of course, again, this is very expensive. We cannot do all of these molecules and we need to do a subset. And that's why artificial intelligence is very, very useful. So in an ad, what we are doing a lot in the lab is we have these molecular modeling techniques that can give us this kind of data. So each point in here will be a different conformation, a different spatial distribution of this ligand within the pocket. And all this data now can be shifted with artificial intelligence in order to train the models to become better. So not only artificial intelligence will be learning one data point per molecule. Let's say we could pass him experimental results. Experimental results will be this molecule works this much, and this other molecule works this other much. So these are just single point data per molecule. But when we do this data augmentation with these simulations, now we can pass him a lot more information, and then artificial intelligence become better. Let me show you an example. This was done with a pharmaceutical company in Spain, Almirai, which is the largest, and they had a different set of molecules that basically were scoring for a target. They had one target. They wanted to find a better drug for this target, a different molecule for this target that could be turned into a drug. And we had this data that they were providing to us, both data in the public space and data in the private space, this validation set that only belonged to Almirai. So now we could use these molecular modeling techniques, the one I showed you before, to extract more data, to do data augmentation, synthetic data. And we did that at very different levels, at the level of just descriptors of the molecule, at the level of using this kind of computational docking predictions, and at the level of now getting our molecule with our techniques and seeing how well it was induced fitting, how were the dynamics of the system in the pocket. And if we had only used the preliminary data, the one that was coming from this distribution of low and high, we could not really reproduce with AI techniques any of the validation set that Almirai had. Basically, it was impossible. But when we expanded the data to all these other techniques, now we got a predictive model that was, correlating quite well. We could really, the predicted top ones were the best ones experimentally. And then we could put this to work, and now we can apply this into a massive screening of some library. And basically, Almirai bought 30 molecules, they could test 20, and they find 2 hits. So they find 2 molecules, the ones in black in here, that were really performing well in their experimental assay. So 2 new molecules that were hits on this thing, which could not be done just by pure AI techniques, given the low amount of data that we had. But when we combined AI techniques with molecular modeling, we could come out with something significantly higher. So now we're doing better. Now we are interrogating, as you saw, these results I showed you before were like for 4 or 5 years ago, where we had like 10 million molecules. Now we can really screen, or we would like to screen. We have accessible 70 billion molecules, which are a lot of molecules. And of course, we cannot do any molecular modeling on those 70 billion. And we need, so these are the on-demand libraries, like really large libraries. We need machine learning to be able to screen very fast in these libraries. But machine learning has the problem that it can score very well the things that are similar to my training set, but it scores quite badly the things that are quite dissimilar, what we call the applicability domain wall. So things that are far away from my training set don't have good predictability. So that's the things that we are applying. It's called active learning. And possibly you heard about active learning in many other fields. It's a common technique. Let's say that we have the traditional molecular modeling, the things that you saw of trying ligand with this molecular modeling descriptions and see whether the score of fitting was going to be better or worse. All right, traditional classical modeling. And that's the first step. So we get a subset of this library. Instead of the 6 billion or 44 billion or 70 billion, we take maybe 100,000, 200,000 or 50K, only a small subset. And now we do the molecular modeling on this small subset. And now we ask machine learning, learn from this synthetic data, learn from this virtual data. It's not that we went into the lab and we try all of them experimentally and we have certainty, but we believe that molecular modeling does okay job. And we tell machine learning to just learn from this molecular modeling. Give me a machine learning model that now I can use this model to very quickly screen very large libraries. And that's what I do. I build a model and then I screen. Now I get the screening results from this machine learning application of the machine learning model, and I don't believe them. I know they're going to be pretty bad at the beginning. So I take the top 50K again, or the top 100K, the best molecules predicted by AI, and I go back into my molecular modeling. So I go back into molecular modeling and I ask for validation. I say, hey, molecular modeling, do you think my predictions are good? And some of them are going to be good, some of them are going to be bad. And this good and bad information is sent back to the machine learning. So it just learns to do better machine learning models. And these machine learning models, again, are used to screen. And we do this kind of iterative cycle. After 3-4 of these iterations, we are getting our desired goal of shifting this distribution. If you remember this movie I showed you before, we have an initial distribution, and we are traveling this distribution towards the left, toward better and better scoring functions, toward values that are extreme, that give me a very good scoring functions that are predicted by my computational techniques to be very good. And that's what you see. Here you see like 4 iterations of this iterative active learning, and you see how it's being shifted towards the left. And each iteration, we are gaining more knowledge of new diversity of molecules. In the bottom plot, you see the decomposition of the chemical matter, of the chemical molecules that I am retrieving that are computationally good, and how they are exploring much more new chemical space. And actually, when we pass, this is actually a real example that we did in our spin-off for a client. We passed them 90 molecules, just at the very tip, the best predicted molecules, and they found 64 hits. So very good hit rate, exceptional hit rate, something that 5 years ago would be unthinkable of. And that was the combination of being able to screen very large libraries, but using artificial intelligence or machine learning to learn from more classical techniques and apply these learnings into our data sets and quickly retrieve from those data sets the best molecules. Now we are doing something similar, but we are doing it with variation autoencoders using generative modeling. So, I mean, all of you are aware of generative modeling, how we can train artificial intelligence with, for example, some images of people, and how we can create new people, virtual people that don't exist, but they have all the features of a normal person, or cats, or dogs, or horses, anything. So the same thing you can do with molecules. You can have a general library, a library of molecules, and you can pass him that information and say, okay, learn from this library how to make new molecules. And then you can have a specific library that's molecules that are good for your target. You want to design a drug for this particular sickness, for this particular target, and you know that these 10, 20, 30 molecules are good for that target. So that's a specific information for my target. So I pass the both of them to a variation autoencoder, a machine learning engine that can tell me can learn, can decode, encode and decode this space and add a little bit of randomness to generate new molecules all the time. And now I can come out with completely new molecules that in principle, I can train them for my specific set. Because of my specific set is very low, I need again to populate it with virtual data. So what I do first is I learn that these molecules are good. So this box here basically is asking for these molecules to be good molecules. Are you, if you're a person, you have two eyes, you have a nose, you have a mouth, you don't have features that are very different from the features I would expect from a molecule. And it just keeps learning, this inner cycle keeps learning to do these things. Once I have enough molecules that are good looking, I activate a second loop of active learning. And now I apply these molecular modeling techniques that will tell me if my molecules are good or bad towards a particular target. And I select those that are going to be good. And the ones that are going to be good, because they have a good score in this molecular modeling, are going to enrich, again, generating virtual data that will enrich my specific set. And I keep doing that. Many inner loop cycles, and then one outer loop cycle. Again, many inner loop cycles and one active loop cycles. So that's the beauty of this active learning, that you can add into them any information. For example, you could have a human sitting right here in some aspects of this outer learner, like for example, right here, and the human being inspecting all the molecules. And only if these molecules he likes them, they will be passing for AI to learn from them. So these are very powerful iterative repetitions that can really teach or can really guide AI into designing a subset of things that fulfill our requirements. And this is actually how it worked for a real system. We just published this in communication chemistry last month. So basically, you are here seeing all the inner loops, how you're learning to make good molecules. And then you say, okay, are these molecules good for my target? Well, half of them are, but half of them are not. So I reduce them, keep accumulating. Yes?
I was wanted to know in the previous slide on the pieces of inner loop, inner active learning and outer active learning.
Yeah.
I'm wondering whether there is any level of fine tuning that is happening on maybe on the variational autoencoder and
No, yeah, not on the variation of the encoder, just on the data training sets. So the only thing we affect is these training sets. That's the only thing we change. We are enriching the specific set that are molecules that are good for my system. We just keep enriching them. So adding molecules that I believe will be better. So we learn from this molecule, he learns from them, and then he gets better. The fine tuning, actually, for example, it's here in the docking score. So every time, if maybe at the beginning we start with a threshold of -6. So we let pass all the molecules that are -6. But because he's learning from those, he will be generating molecules that have better score. So in the next round, we apply a score of -7. And in the next one, a score of -8. So in that sense, we are driving more and more our training set to be better and better molecules. But the variation encoder is as is. It's not being modified.
okay, that raises a few questions more. If the fine tuning happens at the docking, like calculating the docking score,
Yeah.
isn't that like making the model miss out on some of the molecular properties maybe would be important in having or predicting sort of other qualities like binding affinity and maybe the admate of
Yeah,
the
so
molecule?
that's right here. Yeah, you're totally right. And here, actually, we have a lot of admit as well. We can add admit. That's why I was saying you can add anything you want in here. So before we go into an affinity prediction, which will be the docking score, before we go into there, we always ask that in the inner loop that these molecules have good standards. And these standards, for example, involve synthesis accessibility. So these molecules have to be synthesized, or QED, drug-like properties. And as you well said, ADMET, you can add toxicology predictions. You can add similarity or dissimilarity. You can say, okay, every molecule, please be very different from this set, because this set has been already patented and I cannot take advantage of it. Or or the opposite. I want a molecule that looks very much to this initial hit that I have. I just want to decorate it a little bit, make it a little bit changes here and there, and just in that way develop a better structure activity relationship, SAR. So just getting into a better molecules constantly. So you can add anything you want in here. You can add anything, and as you said perfectly well, the first thing that you have to check is that these molecules are good molecules. They don't look disaster, because I can tell you that the first molecules that the BA does are completely disaster.
Perfect. Thank you.
So that's how it is working. So this is actually this was not for a kinase, for a CDK2. We did it for this kinase because this kinase is a heavily populated space. There are already 1,500 molecules described for this kinase. And of course, when you look at generative modeling, you are thinking, oh, I'm going to be able to develop something completely new. I'm going to be very creative here. So we ask for our generative modeling to create molecules that were maximum 0.3 Tanimoto similarity. Tanimoto similarity is an index that goes from 0 to 1, 1 being that the two molecules are identical, 0 being that two molecules are completely different. And below 0.4, the molecules are quite different. So we impose 0.3. So any molecule that the generative modeling was creating, if it was similar to the previous 1,500 published in Campbell, we just throw them to the trash and we ask him to generate a lot of novelty. He was capable of doing that, so we generated molecules that were very different. I mean, that's the two-dimensional decomposition space of molecules. And again, looking at the docking score, how it's traveling towards the left to better and better docking scores. The only thing we are interested in these guys, the 50, 100, 200 molecules that are going to be very good, predicted to be very good. So basically, we selected 10 molecules. We passed these molecules to CRO to experimental synthesis. They told us they could do 6 very easy plus 1 analog. Actually, they told us they could do all of them, but the other 4 were very expensive. So we bought the 7, and actually all of them, all the molecules were active in an experimental assay. Some of them were like around 30 micromolar, one was 1 micromolar, and one of them was 20 nanomolar, like super active. But it was great to see that generative modeling can really generate new molecules, very different, again, maximum 0.3 Tanimoto, very different from the existing space, and all of them being active. And that's actually possibly the technique that we are pushing more in the lab now, like really developing better and better generative modeling. As we said before here, adding more filters, adding more, guiding better the search into a particular subset of molecules, because I think that's really the technique that we will be using in the future, like really designing molecules a la carte, completely specific for our system, etc. And that's interesting because, for example, we all know the pandemic we had, and for example, if we can design a molecule, one antiviral that is effective for a protease of SARS-CoV-2, of SARS and MERS, the last 3 coronavirus, if this molecule is active to the 3 of them, most likely it will be active for the next one coming in 10 years. So that's what we are trying to do right now. And actually, we published a bioarchive recently about this. So we do the same thing, generative modeling, but now when we do the docking score, we tell him to be good for this particular 3 targets, not for 1, but for 3. If you're not good for the 3 targets, we don't like you. And we just keep accumulating molecules that are better and better for these 3 targets. At the beginning, because we are asking 3 targets, we need to relax the threshold. But slowly, you will see in the next slide, we can increase this threshold. And the same way you do it for 3 targets, you can do 3 targets that you want to be docked, and let's say 20, 30, 100 anti-targets, targets that you do not want to touch. And in this way, you can build through these active learning cycles specificity into the system. So this is actually the curving rate. So in the first inner cycle, we had to increase significantly to 40 iterations because, again, you are imposing now 3 targets. So you need to accumulate a lot of molecules, because when you ask if the molecules were good for your target, you decrease it by a lot. But slowly, you see how we can accumulate a lot of new molecules, and we can actually tighten, make the threshold of selecting, which is basically this line here, more and more selective. At the end, we could ask for molecules that were doing better, almost 8.5 glide docking score. So, pretty good predictive molecules. Again, we are shifting the distribution of our docking scores towards the left as we wanted. And the interesting part is that when we are actually clustering all the molecules that we get, we are seeing, depending on different epsilons and dB scan, we are seeing that he keeps learning. So, if we can make this really fast, we can run 20, 30, 40 more iterations and really make, at the end, like the perfect molecule. Of course, now we are working on making this faster so we can really run a lot more cycles and keep learning. So these are the different clusters that we were getting from DB-Scan, showing us that every time we are exploring new spaces, developing new clusters, so the molecules that are coming are significantly different. And that's what we want to do in this kind of polypharmacology workflow. So I believe this can be very interesting in future for pharmacology. Let's imagine a cancer, or let's imagine a neurodegenerative diseases like Alzheimer that depends on very complex factors. And every time we design a drug for it, for example, in cancer, the cancer evolves and is capable of bypassing, developing resistance for our treatment, for our drug. So if we can develop a drug that touches not one, but 2, 3, 4 targets in the therapeutic target profile of this particular disease, then it will be very difficult that this cancer will be evolving accordingly in 4 different targets at the same time. So it's going to have a really, really hard time to overcome these molecules that are acting. It's like you're in a war and suddenly your enemy attacks you not in one front, but in 5 different fronts. So it's very difficult for you to defend out of 5 different fronts attacks. So that's the things that we are doing. In order to do that, we are building a very large library, hundreds of billion compounds. We are applying artificial intelligence techniques to pair these molecules. I'll show you this in a moment. And then we are applying these active learning cycles that, as I showed you before, are acting on multiple targets simultaneously, all right, to develop this polypharmacology score. So the first thing we did is we developed a protein language model to really learn about the pockets of targets. So the pocket of a target is the physical space where the drug will be interacting. And if we want to, let's say in Alzheimer, we have around 217 targets that have been defined in the last
Victor,
potential targets. Yes.
about I think the previous slide where we have the multiple targets and the proteins. Yes. My question is, like, giving some more light about hitting multiple targets. What is happening here, like at a deep level? Like, are we having multiple molecules, or we have a single molecule that would probably talk to multiple protein targets?
A single molecule. That's the beauty of it. A single small molecule that will be specific for your targets, not engaging other targets. And it's not going to be a Frankenstein molecule, like made of 3 different fragments, each fragment targeting one of its pockets. So single molecule, and the only way you can find a single molecule like that is like if you are extremely, amazingly creative. And that's what we are asking generative modeling, to make something that's going to be very, very new, very creative, that will be touching these particular targets, but not the others. So yeah, single molecule. And that way we need generative modeling, or we need a huge library of compounds. We can search really with a huge hundreds of billions to be able to find this very, very particular molecule.
Okay, makes sense. Thank you.
And the first thing we need to is to decide which targets we will be targeting. So let's say in a particular disease, we have hundreds of declared targets. And of course, not all of them are going to be able to be targeted simultaneously by the same molecule. I mean, it will be naive thinking that we just take 2 or 3 targets and now we can design a molecule that will bind into those. I mean, that's not going to happen, or at least I don't imagine how that could happen. But we can ask machine learning, a language model, to tell us which pockets will be able to be targeted simultaneously. And the way of doing that is we have done a multimodal embedding, one with sequence space of the proteins, like typical protein language models, and another one with a structural space. And we have trained that with all the database of AlphaFold known pockets, all right? So now we have an algorithm that can really find pockets and has a lot of information of these pockets. In order to find them, it's because you can have a lot of descriptors, a lot of inner information relating not only sequence, but sequence and structural motifs, distances between amino acids in the pocket space. We actually presented Pickpocket last April in Singapore in the ICLR 2025, so one of the best learning representation conferences in the world. And we could show how Pickpocket, overall, with recall and precision, is at the top of the binding pockets predictors. And we need that because we are not actually asking to find a binding pocket. We are asking him to correlate pockets for polypharmacology. So we need to be really precise. We need to really gather a lot of very good information of the pockets. So now let's say we have this superior technique to find out pockets and to analyze information of pockets. Now we can train a Siamese network, a dual Siamese network, where we have two, we did this in a binary way, a pairwise way. So now we have two proteins. We have the language model that will be describing the pockets in this pocket, in these proteins. And now we train it with all the data that we know of polypharmacology. And we are creating a distance space. So this Siamese network just computes the distance. How far are these two pockets in the druggability space? So pockets that are really far away, they won't be able to be drugged by the same molecule. And pockets that are closer, they're going to be able to be drugged by the same molecule. So we took around 30 random proteins from a data set of proteins, and we asked him to pair some of them. And he told us, okay, these are, I believe, machine learning told me, these are in dark blue, the proteins that I believe can be targeted by the same molecule. And these are the proteins that I don't think they can be targeted by the same molecule. And I can tell you that within these proteins, there was not similarity between them. So they were quite different proteins. Then we took 5,000 molecules from the enamine space, and we docked these 5,000 molecules to each of these pairs, the ones that are predicted to be polypharmacology and the ones that are predicted to be non-polypharmacology. And we can see how the ones that were predicted in darker blue have significantly better average docking scores than the ones that are not predicted. So it's very nice to see how something that has been trained fully on the sequence and structural space of pockets can really correlate well with docking scores that are more informative of the molecular complementarity of molecules. So, we believe now that we have a technique to really pair up targets to tell us, okay, out of these 100 targets, maybe these 4 can be drugged simultaneously, again, by one single small molecule. And then we need to make this fast. We need to compute, we need to interrogate hundreds of billions of molecules or generate a lot of molecules in a fast manner, because I anticipate that's going to be very difficult to find such a molecule. So we are also working in a multimodal language model to predict drug affinity, similarly to what isomorphic labs and many others are doing. And we are having a sequence space encoding, a molecular space encoding, and our pickpocket that I showed you before. And we have the 3 of them and has been training in around 16 million data points of ligand affinity. And now we can very quickly screen molecules for a target, and we can see how, again, in a distance similarity, it can group up molecules that are known to interact with one target can be grouped in the vicinity of target. And this takes fractions of milliseconds. So we can now screen very large libraries and be really fast. Because what we want at the end is have these active learning cycles, having a machine learning scoring function, but our affinity prediction, we want to change the molecular modeling affinity prediction by a faster artificial intelligence affinity prediction. So we can very quickly score or predict hundreds of billions of molecules, or we can very quickly apply it into a generative modeling scheme. And these are the things that this is really work going on in the lab right now. Let me show you a little bit, just to finalize, how we are applying also these active learning cycles, not for a small molecule, but for biologics, for larger constructions of molecules, like antibodies, or for protein-protein predictions, etc. So again, we have, yes?
Yes, before maybe you conclude, on the previous slides, you've talked about the protein interaction, and I think ligand protein interaction, but also somewhere you talked about the pockets. And I'm wondering, is there any special way you've encoded ligand features or even pocket features in pickpocket, if that was talked about anyway?
Yeah, so in this multimodal representation, now we have ligand features. So we take, for example, a solubility prediction that encodes millions of molecules solubility prediction. So now we have a language that speaks the molecule, or an embedding that speaks the molecule language. All right, not our affinity, but at least learns the representation of molecules and learns what is important in different molecules. So we have a protein language, a molecular language, and a pocket language. And now we are combining the three of them with data that is specific for protein ligand affinity. So that way we can train this multimodal embedding into something that will speak the protein ligand affinity language. That's our goal. Does this respond to the question?
Yeah, yes, sure.
So yeah, you need, as you said, you need to have all these embeddings. You need to have all these different learning representations. Otherwise, it's going to be impossible. Yeah.
Thank you.
And similar things we can do for larger groups, for biologics. Here in biologics, you're going to be talking about proteins, so you are going to have a lot of evolution space, like a lot of sequence alignment, and the same way that AlphaFold is mostly based in descriptors that are mapping the evolution space within proteins. We can also have diffusion models that will be creating completely new molecules. We'll be collapsing the backbone of a protein that will be better fitting a protein-protein interface, for example. So we have all these machine learning techniques in this side here. And then we have, again, the molecular modeling techniques that will be telling me whether a design that's coming from AI, at least from the molecular modeling perspective, it's good or bad. And again, we are sharing information. We are trying to guide AI into a better space, etc., etc. Let me show you some examples. For example, again, generative modeling of proteins. So instead of generating small molecule drugs, here we're generating full proteins. And we did that with a company in Germany, it's called Navigo Proteins, for finding ubiquitin-like proteins that were binding into a target, HER3. Actually, we published this last year. The work was done 2 years ago, but actually the protein that we find for them was patented and was, the process of patenting took quite long, and it took quite a while for us to publish it. Again, we are doing these kind of active learning cycles where we are generating sequences, we are classifying sequences, and then we are using molecular modeling to discriminate sequences, to tell us whether this sequence is good or bad, and then do the design again, etc., etc. And this is, for example, the numbers that this company, Navigo Protein, was telling us, experimental numbers. So for them, it took around 6 months to generate experimentally the sequence and to try them in the lab. They assayed around 100,000 sequences, so new constructs of proteins. I don't know exactly the cost. They didn't disclose the cost, but I can imagine this will go on the hundreds of thousands of euros. And at the end, they found 12 sequences that were binding, and 4 of them with KD better than 100 nanomolar. We went into the lab, into the computational lab, into the computer. It took us 1 to 2 months. Now we could do it in 1 month. It was the first time we did it, so probably that's why it took us a little bit longer. We provide them 435 new sequences. They only tried 12 experimentally. Actually, out of these 12, 7 were binders and 2 had KD, desired KD affinity to our protein below 100 nanomolar. So significantly better results at a much, much cheaper rate and faster results. So this is what this combination, this active learning going from AI to molecular modeling can give us in this space. And for example, let me show you an example. This is vaccine development. So we are heavily involved in vaccine development because these artificial intelligence techniques can very quickly now provide new proteins and tell us whether a protein will be interacting with another one. So let's say for a vaccine development, basically what we want is we want to optimize a protein, for example, a protein of the virus or a protein of the pathogen that we want to vaccinate against. And we want to take the green area, which is the epitope that has really the activity that will make it pathogen, for example, binding towards our cellular cell. This green area will bind into our cellular cell and will promote the virus entrance into the cell. So we want to take this green area and we want to optimize it really well, the reminder. So the green we cannot touch because it has to be recognized by our immune system. But everything that's gray, we can design new proteins that will be making very, very, very stable vaccines, very stable epitopes or proteins to be vaccinated that will be eliciting a very high immunogenic response into the body. So what we need is to make very, very strong epitopes that we can use for vaccines. So the way we do it is with machine learning, like diffusion techniques or protein PNN, which is a learning representation of inverse folding that can predict new sequences, techniques that have been developed, for example, for the last Nobel Prize, David Baker, but not the last, the previous Nobel Prize of last year, and that will be inserting new sequences in our protein that are going to be predicted to be more stable. Then once we have these things, we have to eliminate glycosylation sites. So we don't want to add new glycosylation in the proteins that will be masking, for example, the epitope recognition by the immune system. Now, when we have all these potential new sequences, we use different these different folding techniques, co-folding techniques, like AlphaFold or ESM fold. And we impose that the two of them give the similar answer. If the two answers is below 3 angstroms, then we like it. If it's above 3 angstroms, we discard it. Then we compute cross-reactivity. We don't want to insert anything in the body that will be having an autoimmune response. And all the final candidates, we do a better prediction of the structure, and we do a lot of molecular dynamic simulations to make sure that this is a very, very stable protein. So we have done already 3 vaccines. One of them was COVID, long time ago. Actually, we patented the vaccine. And now more recently, we have done a vaccine with a Spanish company for a respiratory virus. So all these designs were AI-based. So all the vaccines that we passed them to them were artificial intelligence designs that fulfill this platform that this workflow that I just showed you. These are the in vitro results. So we were getting all our designs are in light green, comparing to the known vaccines or the vaccines that are in clinical phases, more or less similar result. And actually now we have a second generation that's a little bit higher. And when we went into mice results, we can see that our designs were eliciting a very good response in mice. So actually this vaccine is going to go now into clinical trials in humans. And then also we have now a European consortium for the West Nile virus. Here we are not as advanced. Here what we have done is designing our protein expressions or protein constructs that can have a very, very good expression. Our proteins that are very solid, very stable. So that's basically the baseline expression that we had. And actually this was already a little bit improved by our experimental coworkers. So we have been able to develop a protein that expresses 5 times better. So it's much more stable and most likely will elicit a much more larger immunogenic response. And basically, we did a second round, and actually we improved this a little bit more, almost 6 times better expression. And now these constructs are entering as of December, like in a month, we'll be starting mice inoculation to see how well these vaccines will work on mice. But I can predict that in 1 year, year and a half, or maximum 2 years, we're going to have a very good clinical candidate for West Nile virus vaccination. Antibody pipeline, very similarly, we can develop better antibodies. We use, again, AI de novo generation of antibodies, and then we are applying all these molecular modeling techniques to filter them, and we can generate antibodies that have better binding affinities or that are more specific for a given epitope, so we can really antigen, so we can really develop a new flow of antibodies. And then I think is going to be really the one of the next revolutions in pharmacology, how with AI, and in particular combining molecular modeling with AI, we can really generate very, very specific and better working antibodies. And this will be, one of the, we are already assisting to a lot of new companies, new biotechs being formed by basically this type of combinations. So, all right. So for conclusions, AI is here to stay. AI is already making a huge impact, already designing better vaccines, already helping us to screen through active learning processes, large libraries with more efficient interactions. And, if we can now have a full AI by changing the molecular modeling reinforcement, all these things that are using more traditional techniques, if we can change that for better, faster AI predictions, we can have something that can really work in the hours, in the days, and give us a very, very quick solution to a critical, urgent pharmacological matter. So let me just thank the people that are at the computational at the company in Nostrum, and also at the academic site at Barcelona Supercomputing Center, and also all of you for your attention to this talk.
Thank you, Victor. Mine is just a question of curiosity. You talked about the sort of evaluation that happened where you supplied, I think, 430 something, and then 12 sort of proteins, and then somewhere you said 7 were, I think, targeting, were sort of like docking. And the surprising question, yes, is about the computation time that was taken. Yes, we see 6 months, but also the 1 to 2 months is also still scaring, given the computing resources that you have, and what explains that time?
So that was the first time we did it. So we had to develop everything, and that's why it took 1 to 2 months. Now we can do this probably in around 3 weeks, I would say. And of course, it just depends on having more access to computation. But again, it's an active learning, so you need to run several cycles. So one cycle might take 2 to 3 days, but then you need a little bit of human intervention to look at the things, etc. Right now, for example, this workflow of vaccine development, we can run it in 4 days. And actually, we are going to be part of a national security program that's going to be designing, hopefully, if we get the grant, designing a national security platform to, under the risk of a serious invasion of viruses, etc., we can design a computational vaccine in less than a week. And that's
Wow.
what AI plus, of course, heavy usage of computation can provide to you. But of course, then still needs to be tried in the labs, which is going to take another month. So but I mean, COVID has been really great in science, in how we are doing things now, and now giving a new virus, I think we can probably come out with a vaccine in very few months, which was something that was unthinkable of before 2020.
I think that's amazing work, and the type of work that you're doing in Barcelona. It's really inspiring, and I'm really impressed. I'm happy that we are seeing the link to the code, I think. And I think I
Yes.
would encourage everyone to look it up and check it out. If there is anyone in the audience with a question, please, you can unmute yourself And if there is no, I'd like to conclude this session by thanking Victor. He refused, stopped me from calling him professor, but thank you very much. It is really an inspiring session and great work that is novel. The paper is published in Nature, and you guys, you could check it out. Thank you very much, Professor Victor. Thank you very much indeed. And yeah,
Thank you for the invitation. And if you guys have any questions, feel free to reach me by email anytime.